Okay, so this is about the deviations from Mendel inheritance. So we have 6 deviations from the Mendelian inheritance: codominant alleles, incomplete dominant alleles, multiple alleles, linked gene, sex-linked genes, polygenes. Okay, so this video I'm going to focus on linked gene first. So what means deviation from Mendelian inheritance? If you still remember that previously we have learned about the Mendel first law and second law, we say that monohybrid cross, the law of segregation for Mendel first law, we should get this ratio, for example 3:1, 1:1, and 1:1. Dihybrid cross, we should get 9:3:1 or 1:1:1:1 for the test cross. So all these deviation from the Mendel law, meaning that we will not get the following crosses. Okay, we will not get the following ratio. Okay, so this is called deviation from the Mendel law. Alright, so I'm going to talk about the linked gene as it is quite related with our dihybrid cross. Okay, so first of all, what means linked gene? Okay, link meaning that join. So a condition when two characteristics, each controlled by different gene, located on the same chromosome. So it's quite similar with our dihybrid cross. Because dihybrid cross, it's also control two characteristics, controlled by two different genes, but the difference is dihybrid gene is located on the different chromosome. For this linked gene, they are located on the same chromosome. So therefore, linked gene do not obey Mendel's law. Okay? Why they do not obey Mendel's law? Because they cannot undergo independent assortment. The genes inherited together unless they are separated by crossing over during prophase 1 of meiosis. So for example, you can look at the diagram here. For the left hand side, it is a diagram of dihybrid cross. Okay, for dihybrid cross, we have gene A and gene B. Okay, it's located on different chromosomes. So when they form the gamete during the process of meiosis 1 and meiosis 2, so they can assort independently to form 4 different possibilities of the gametes: the big A, big B, small a, big B, big A, small b, and small a, small b. Okay, without crossing over. But for the linked gene case, linked gene means the gene A and gene B is located on the same chromosome. So if without crossing over, they can only form the gamete which is big A, big B, and big A, big B. The other one, they form the small a, small b, and the small a, small b. So the possibility of the formation of gametes will be 50% lesser compared to dihybrid cross. Okay, dihybrid cross, you have 4 different possibilities of gametes, but for linked gene, without crossing over, you only get 2 possibilities of the gametes. Alright, so therefore, linked gene cannot get the result as dihybrid cross, although both of them also contain 2 genes, gene A and gene B. Okay, however, if the linked gene cases, gene A and gene B, they undergo the process of crossing over during the process of prophase 1. Okay, so crossing over, they exchange the genetic materials. So there is the possibility of the linked gene to produce 4 different possibilities of the gamete as well. For example here, crossing over occurs here. So now they produce the gamete big A, big B. So this one, after exchange, they produce small a, big B. This one after exchange, they produce big A, small b, and this one is small a, small b. Alright, so this big A, big B, and small a, small b, we call it as parental gametes. Okay, it's that exactly the same like the parent. So this small a, big B, and big A, small b after exchange, you call it as recombinant gametes. Recombinant means it is a new combination. It never appeared from the parent generation before. So there is 4 possibilities of the gametes if crossing over occur for the case of linked gene. Okay, so let's see one of the example of the questions here. A Drosophila has dominant gene for gray body and normal wing. Okay, so they gave you the letter gray body, capital letter G. So understood that gray body is dominant. Normal wing is capital letter V, so normal wing is also dominant. So the recessive allele for both of the gene are black body and vestigial wings. So the flies with heterozygous gray body and normal wing is crossed with black body and vestigial wing fly. So the results, they gave you the following results here. So first we analyze the questions. The characteristics for this Drosophila fly has 2. First one is the color of the body. Color is either gray color or black color. So gray color is dominant, black color is recessive. Second one is the shape of the wing. So the shape of the wing, we have 2. There is either a normal wing or vestigial wing. So normal wing is dominant, okay, while vestigial wing is recessive. So when 2 characteristics involved, we should always consider, are they dihybrid gene or are they linked gene? Okay, so see the following questions now. The fly with heterozygous gray body and normal wing. So heterozygous means that it is a cross of big G, small g, big V, small v with, okay, black body, vestigial wing. So black body, vestigial wing, both of them are recessive. So it's a cross between small g, small v, small v. So indirectly, they told you that this is a type of test cross. So if the dihybrid cross test cross, as we have learned before, the results for dihybrid test cross should follow Mendel's inheritance law, that is, produce 1, 1, 1, 1. But we see the results here. 236, 53, 250, and 61. So are they 1, 1, 1, 1? Definitely is not. Okay, so the question asks, what is the type of crossing here? So you cannot answer as dihybrid test cross. It is not dihybrid test cross because based on the results here. So what can we answer here? You can say that, okay, they are a type of linked gene. Okay, and test cross. So what can you conclude about the genes in this inheritance? We can say that these genes are linked, okay, or the 2 genes are located on the same chromosome. So show the crossing diagram. Okay, so now I'm going to show you how should we draw the linked gene genetic diagram. It is slightly different from the dihybrid cross. Okay, so as we know, dihybrid cross. Okay, dihybrid cross, they involve 2 genes, A and B, both on different chromosomes. So we can apply the law of independent assortment. So during the formation of the gametes, you can form either big A, big B, small a, small b, or big A, small b, or big small a, big B. So this is independent assortment. They can assort independently without anything control them. Alright, so therefore the test cross, you do it like this: big A, big B, small b, cross with small a, small b, small b, formation of the gamete, four of the possibilities. For this one, only one possibility. So your F1 generation with 4 different genotype, phenotype and genotype ratio is 1 to 1 to 1 to 1. Okay, so that's the normal dihybrid test cross. So what's the difference with the linked gene test cross? So linked gene, like I said just now, it consists of 2 genes as well, but both of them are located on the same chromosome. So if without crossing over, they form only 2 possibilities of the gametes, big A, big B, or small a, small b. Okay, but if crossing over occur during prophase 1, okay, like I said just now, they exchange genetic material here. So you form 4 possibilities of the gamete, big A, big B, big A, small b, small a, big B, small a, small b. So this is how we construct the genetic diagram for the linked gene test cross. So did you notice that the way I write the genotype is slightly different from the way I write for dihybrid cross? We should write big A, big B / small a, small b, not big A, big B, small b. Why is it so? Because we want to show that AB is located on the same chromosome. Okay, AB located on the same chromosome / another one is from another chromosome. Okay, a pair of homologous chromosome here. So since this is test cross, so we cross with a homozygous recessive case. So for the gametes here, crossing over occurs. So you form 4 possibilities of the gametes here. For this one, since they are homozygous, so no longer no matter they cross over or do not cross over, they only have one type of gametes. So the F1 here, you write the genotype, okay, like this, means that they are on the same chromosome, big A, big B, so this one is small a, small b, big A, small b, and this one small a, small b, small a, big B, so this one small a, small b, and this one small a, small b, and this one small a, small b. So we can see that this big A, big B, small a, small b is like this parent. So you call this parental genotype. This small a, small b, small a, small b, same like this one. So this is also parental genotype. So this is something different from the parent. So you call it as recombinant genotype. So the ratio from the test cross is not 1, 1, 1, 1. Okay, not like the ratio here. So what should be the ratio for the test cross here? Okay, so you can refer back to here. We write it like this. Okay, how many of them gray normal? How many of them black vestigial, gray vestigial, and black normal? So use the value given here, 236, 250, 53, and also 61. Okay, it's not 1, 1, 1, 1. You cannot simplify them into a simple ratio. So you can see that we usually have 2 groups with a larger number, 2 groups with a smaller number. So this is the trend for linked gene test cross. So this larger number, we say that they are parental genotype and phenotype. Smaller number is a recombinant genotype and phenotype. Okay. So you also can see that the way that gamete is written, you can also draw it like this, or using the way which I show you just now. Alright.